Hi, Bio245. So we're going to be talking about muscle fiber on the model, so that you can actually identify the various parts on this model that people view as very confusing, but really it's very easy. I think the one part where people really get tripped up is that they have a very difficult time envisioning this as a cell. So first, I just want to show you real quick that we do have corresponding components that are the exact same. They're called a little bit different, but they're the same exact thing. Okay, just whenever you have muscle physiologists, they all come up with their own special names because they're jerks. So it's just the way it goes. So first off, here's our cell, just kind of crudely drawn. You can see here I have a nucleus indicated with this dashed black line for our nuclear envelope. Inside of that nucleus, we have a nucleolus, life is good. We have the endoplasmic reticulum, and I just made it as smooth just for clarity. But we have our endoplasmic reticulum here outlined in this baby blue color. And we also have some mitochondria that are colored in red. I even indicated just a Golgi in here just for fun. But anyway, so there's our basic components, right? Nucleus, endoplasmic reticulum, mitochondria, and of course our plasma membrane, right? So that's a cell. This muscle thing, this is a cell too. It's just organized a little differently, okay? So I wanted to highlight the fact that in the case of, on this particular model, and we'll talk about this, but I just want to show you, see this like webbing kind of stuff? Anyway, I have that drawn in blue here. So this stuff is this cell's version of an endoplasmic reticulum. It just has a different name, and we'll talk about that. And also, it has all these little mitochondria that are shoved off to the side. It also has multiple nuclei, which I've indicated with the dashed black line and the nucleolus inside. So, same exact cellular components. This is a cell. It just looks different and is organized a little differently. So, now that we've got that out of the way, let's talk about our muscle fiber. So the first thing I want to do is kind of harken back to our part 1 video of the sliding filament theory. Remember that when we take our muscle and we do a cross-section of it, we see those fascicles that are surrounded by perimysium, and that we can then parse that out a little bit more to take out our individual muscle cells or muscle fibers that they're surrounded by endomysium. So, and then also, like I mentioned before, you can take that muscle cell apart even further and take out those tightly packed bundles of protein called myofibrils. So let's turn this guy over real quick because I want to show you what's going on here. Let's do it like this so you can actually see. There we go. So here's our muscle fiber cross-section. Here's our densely packed bundles of protein referred to as myofibrils. Okay, so that's what we're looking at here. Remember how I said that a cell cytoskeleton is just kind of like this protein network that helps to help the cell keep shape and help ship stuff around. And remember how I told you how that muscle fiber, they just more tightly pack those proteins together into these long tubular structures. There you go. So you can see like that we have these long tubular structures, and all that is protein. It's all actin and tropomyosin, troponin, and myosin, just tightly packed together into these tubular shapes. So when we have these other cellular organelles that we need to shove into this model, it winds up just being oriented around the actual myofibrils. The myofibrils take up the majority of the cellular compartment. So let's talk a little bit about those organelles first so you can see how they're the same. So here's our plasma membrane on the outside of the cell, same exact ordeal as we see with a normal cell. And just to show you as well, remember how I said that we have that epimysium on the outside of the muscle, perimysium on the outside of the fascicle, endomysium on the outside of each individual cell. Here is our endomysium on the outside of our muscle cell. Okay, so there's our endomysium, just a layer of connective tissue. And again, dense, irregular, so that's why you see all this crisscrossing fibers irregularly placed with respect to one another. So we have our plasma membrane. You'll also notice that we have little baby mitochondria and that they are kind of strewn about the cell. So there it is. And you can even see that there's that cross section. Remember how I told you whenever you have a mitochondria and you cut it open that you'll see those cisternae? Booyah, right there. Okay? So there's our mitochondria. The other thing you're going to see is endoplasmic reticulum, but on the muscle fiber, it's shaped a little differently because in a regular round cell, the endoplasmic reticulum kind of folds out throughout the cytoplasm. It doesn't have those proteins that it needs to really worry about. They're not densely packed. They're kind of loose and hanging around the cytoplasm. But in the case of our muscle cell, we have these densely packed protein fibers. So the way the endoplasmic reticulum works out spatially is it just wraps around each and every myofibril. So here's a myofibril wrapped with this endoplasmic reticulum, and it's this beige veiny stuff. Sorry, it's like inverted, so I'm like not knowing where I'm pointing. But it's this beige veiny stuff, and it surrounds each and every myofibril. Okay, so here's a myofibril, here's a myofibril, here's a myofibril, and here's a myofibril. And what they call it in muscle instead of endoplasmic reticulum is a sarcoplasmic reticulum. Sarcoplasmic. So as long as you remember the word endoplasmic reticulum, just tack on sarco instead of endo, and you're good to go. Okay, so that's the sarcoplasmic reticulum. You'll recall that whenever we have muscle, we call stuff sarco. The other thing that you're going to see in here are some nuclei. So if you look at this long purple thing here, that's supposed to be the nucleus, one of many for this particular cell. The other thing that you're going to see on here is not a normal cell structure, but you'll probably immediately notice, really, these blue tubes right here. Okay, these blue tubes surround each and every single myofibril. So here's a myofibril, here's a myofibril, and you can see that that blue tube encompasses that myofibril. Okay. These blue tubes are just called T-tubules. So T stands for transverse because obviously the muscle cell is running this way. The T-tubules run transverse to that, so it's a transverse tubule, T-tubule. Okay. So anyway, what these guys are doing is they act as an infolding of the plasma membrane. So see how there's like this hole right here, and it goes into this tube. It's an actual extension of that plasma membrane. The reason for this, if you remember our excitation, contraction, coupling, and our sliding filament theory stuff, that we have excitation occur, the muscle cell becomes excited, and then it releases calcium from the sarcoplasmic reticulum, endoplasmic reticulum, but in this case specifically called the sarcoplasmic reticulum, right? Now, if you have a myofibril that is deep inside of the cell and far away from our plasma membrane, remember, the plasma membrane is where we have those acetylcholine receptors, and that is what ultimately results in excitation of the cell because that's sodium, right? Well, if you got one dude who's all the way inside of here, how is he supposed to get the message from the plasma membrane, right? How is he supposed to know that excitation's happened? He's inside like, I don't know what's going on. So what he needs is a T-tubule that wraps all the way around him, so that way when sodium floods in, it floods into the T-tubule, down these tubes, and again, you can even see that they're very stacked all the way up along there, and it wraps around every single myofibril and delivers the message of excitation even deep into the inside of the cell. That way we have solid contraction along from the outside to the inside of the cell, and not the outside contracting and then eventually followed by the inside of the cell contracting. So that's why we got our T-tubules. Okay, what else do we have here? So that's basically our parts of the cell. You'll also notice that there's this weirdo stuff going on the outside. First off, this right here is our axon for our neuron that is communicating with our muscle cell. You'll remember that from our excitation, contraction, coupling video. So that axon goes all the way up here, and here is our axon terminal, right in here. And you see all these little white ball structures? These white ball structures are the vesicles that are filled with acetylcholine. Cool, right? So it's exactly like that drawing I showed you. Here's our little axon terminal, and each of these is filled with acetylcholine. So when that action potential travels down the axon, boom, we get release of acetylcholine, and it fills, you guys should remember, the synaptic cleft, right? So the space between the muscle fiber and the axon terminal, that's our synaptic cleft. And you'll recall when I was telling you about the motor end plate, how I said that on our model is going to be represented by this kind of wrinkly plasma membrane. Well, here it is. Here's our wrinkly plasma membrane, and you can see it's the same color as our plasma membrane out here. That way you know it's still plasma membrane. That is our motor end plate, and it is studded with those acetylcholine receptors. Okay? And again, this is our neuromuscular junction. Here's our neuron, here's our muscle, they're interacting together. Life is happy. Okay? But I believe that's really it for our muscle cell model. So just to recap real quick, we got plasma membrane, we got sarcoplasmic reticulum right here, that webby veiny stuff, which is just endoplasmic reticulum, just specially shaped and named a little bit differently. We have our T-tubules, which are just infoldings of our plasma membrane to help deliver that excitation from the outside in. We have our endomysium, which is this layer of connective tissue that surrounds each and every single individual cell. We have our neuromuscular junction, so we have the neural aspect and the muscular aspect. We have the axon terminal, We have acetylcholine in its little beads. We have the synaptic cleft. We have the motor end plate overall at neuromuscular junction. And I think I forgot to mention that you can also, like I said, see all these mitochondria tightly packed in there. Okay? And also, of course, one of our many nuclei. Okay? And that is it.